Okay, good. Good. All right, so what we're about to do is show you the cellular respiration flowchart. And if you walked in and you saw what you saw, you were probably like, oh my God, yeah, it is. And that's why I put a lot of pressure on you guys. You need to learn this. Because without this process, you're not sitting in that chair of yours. You're not allowed, you can't survive. Because what's the main purpose of cellular respiration is to make what? ATP. So it all starts out with, here's the cell membrane. I have a nice carrier or transporter protein in there. It's an integral protein. And it's working very hard. Sometimes I have to look for these things. Come on. It is working very hard to transport glucose into the cell. So here's glucose entering into the cell by facilitated diffusion. For the most part, it's facilitated diffusion. In certain types of cells, it might be active transport. But we'll just say facilitated diffusion. When you go to A&P, they'll teach you all the other things. So glucose comes in, and now it's inside the cell. So from this point on, that's inside the cell. Specifically, the cytoplasm. Because inside the cell could also mean the nucleus. No, it doesn't go to the nucleus. It's in the cytoplasm. So that means, to orient ourselves, that means, where's my other one? Come on, guy. Intracellular. I'll find it. Intracellular is here. That means inside the cell. It's hiding underneath one of them. Here's the extracellular, outside of the cell. So we're orienting ourselves, intracellular and extracellular. But now the glucose, which obviously you can see is a 6-carbon sugar. So it's a what ose? A pentose? A triose? A hexose? What is it? It's a hexose. It is now in the cytoplasm and it's about to break down. And when it breaks down, it's going to release electrons. The electrons have to be loaded on that truck. Do you remember that? There are two kinds of trucks. There's the NADH truck, and there's the FADH2 loaded truck. But before they get loaded, these are the loaded ones. The reduced ones are NADH or FADH2. But the empty truck is NAD plus or FAD. Got it? I hope when you go move from one place to the other, you don't rent a loaded truck because you just wasted your money. Where are you going to put your furniture in? So you rent an empty truck. So an empty truck will come. Those are the oxidized ones. And load with the electrons. And what do delivery trucks do? They deliver. It's not a tricky question. They deliver. So that means these guys are going to deliver their cargo to someone. And what is the cargo? Electrons and hydrogen. Because to get this, you're going to need 2 electrons, 2 hydrogens, and you'll end up with that. Here too, you have, I forgot to put that in there, but you need electrons and hydrogen for that one too. So you need electrons and hydrogen. So that's what they're loaded for. Good. So that means electrons are coming off of glucose. Yeah. We're going to load them on the trucks. Yeah. The trucks are going to go somewhere to deliver them. Of course. And that's when the fun begins. Wherever these guys are going, that's where the fun begins. That's where most of your ATP is going to be made. And that's why cellular respiration is a little complicated. Not really the first part. It's when these guys get involved, then it becomes really overwhelming, but you can do it. So now we start out with the glucose. Of course, hopefully you realize that's the cell membrane. The glucose is going to break down to these guys. Glyceraldehyde 3-phosphate, dihydroxyacetone phosphate. And then the dihydroxyacetone phosphate will convert to glyceraldehyde 3-phosphate, and you can do that back and forth. So technically, you're making two of these. So what did you just do? You took a 6-carbon molecule here, 1, 2, 3, 4, 5, 6. You see that? Which is glucose. And you broke it into 2 G3Ps. Did you lose any carbon? No. No carbons were lost. That is called glycolysis. Breaking down sugar, lysis, breaking down sugar. Where does it happen? In the cytoplasm. And that's what I was telling you guys. If you know the chart, you know all the answers to the exam that has to do with this topic. Because one of the questions on the exam, and I'm almost certain it's on there, where does glycolysis occur? Where? That means location. Where? It's in the side of, do you see it on the board? Yeah. And once I'm done, all the answers to the question that's related to cellular respiration on exam 3 will be on the board. Your job is to retain it. And when do you start doing that? When? We said yesterday, right? We said actually 2 days ago. But in case you haven't started, it's time to start. It's one thing understanding what's going on. It's another thing realizing that it's complicated. It's a completely different volume to say, I have to retain it. Multiple exposure will help you retain it. So that you can get a mental image of the flowchart. I actually can close my eye, I can see it right in front of me. Really weird. Well, why? Because I looked at it so many times. So that you have to do. In the beginning, there's what's called an energy investment phase. That means you are going to use energy to get the process going. And anytime we say energy, what are we talking about? ATP. On this side is using. So we're going to bring in not just one ATP, actually 2 ATPs. What's going to come out is 2 ADPs. Every time you use an ATP, an ADP comes out. And every time you consume ADP, an ATP is produced if you're adding a phosphate. This is not the entire process of glycolysis. This is only the beginning. Because it's still, glycolysis is up to pyruvate. We don't have pyruvate yet. This is equivalent to when I told you, when you go have a barbecue, what do you need to bring with you? Some people said beer, right? Barbecue sauce. But none of that would be helpful until you have a lighter. If you're going to make a barbecue, you better have a lighter. Otherwise, you'll have raw meat. No one likes that. That's your lighter. That's your spark. Give me a spark. I'll give you a fire. The rest of it is your fire. And that's the spark. 2 ATPs. I'll give you 38. That's a deal. If I tell you give me $2, I'll give you 38, you better take me up on it because you made money. All right? It's the same thing here. So that's what's called the energy investment phase. But glycolysis itself is not done. We have to take glyceraldehyde 3-phosphate down to pyruvate. Come on, pyruvate. Where is it? Come on. It should be right in front of me here. Right in front of me. And pyruvate. Here they are. That is called the energy payoff phase. That means we're producing energy in the form of what again? ATP. And to produce ATP, we have to use ADP. For each pyruvate, you are using 2 of these. And producing 2 ATPs. On this side are the things being used. On this side are the things being produced. Everybody okay? So if we tally things up, we're going to find that we made 4 ATPs so far. Do you see them? But how many did we use? 2. So we made 4 ATPs. By what process? SLP. I'll explain to you what that is again. But we used 2 ATPs. If you're an accountant, you would then say, therefore the net gain is how many? 2 ATPs. And they're made by SLP. And SLP stands for substrate level phosphorylation. We'll put that information up there. Substrate level phosphorylation is this. If I'm a substrate with a phosphate, here I am. If you are ADP and I want to make you ATP, all I have to do is give you the phosphate. I'll give you the phosphate. You become ATP. What do I become? A product. Who cares? What's more important is you became ATP. These 4 are made that way. I wish all 38 were made that way. We'd be here 5 minutes and we're done. But they're not. I'm going to show you. Now, these 4 are made that way, but the net is 2. I'm going to show you 2 more made that way. So out of the 38 ATPs, only 4 are made that way. 34 are made the other way. Which other way is that? We'll talk about it. And it's that other way that drives students crazy. Because this is pretty straightforward. Once it goes into the mitochondria, everything goes out the window. All right? But we'll deal with it, and we'll figure it out. Now, this is glycolysis. This happens whether you have oxygen or not. It is not dependent on oxygen anyway. Do you see oxygen there? So whether you're breathing or not, you will do this. Now, let's use the scenario where oxygen is not present. Let's say oxygen is not present. What's going to happen to the pyruvates? Well, let's see. Both pyruvates will undergo something called fermentation. Oxygen not present. The pyruvates will ferment. Ferment into what? Well, it depends on who you are. If you're an animal cell, and that's what we are, yeah, sorry, we're animals there. you're going to produce lactic acid. And that's what gives you the ouch in your muscles when you exercise. If you are yeast, thank you for that, yeast, you make our wine and our beer. You make ethanol when you ferment. If you were bacteria, it depends on who you are. It's variable. Some bacteria produce lactic acid. Some bacteria produce ethanol. Some bacteria produce a mixture of things. It depends on which bacteria. So we just say variable. When you go to micro, you'll learn more. But then we have to tally things up. There's one other thing, by the way, with glycolysis. We forgot to mention the trucks. In glycolysis, we also load trucks. At this step here, we're loading trucks. The electrons coming off of this get loaded onto NADH. So if we tally things up at the end of glycolysis, we have a net gain of 2 ATPs. Do you see them? 4 here, 2 here. Those are made, those are used. Net gain is 2. But we also have 2 NADHs. The loaded trucks. Loaded with what? Electrons and hydrogen. Now, listen to it carefully. Very critical part. Without oxygen, your cell is only surviving on these 2 ATPs. That's it. Good luck with that. We can't survive that way. Because you have a beating heart, a working brain, active transport all over the place, vesicles being transported everywhere. You need energy. You need a lot of energy. This will not sustain your life. Some bacteria, that's enough. But not for us. We are not efficient without oxygen. That's why you have to breathe it. Because as you'll see, with oxygen, this does not happen. Something else happens. But we're right now talking about the situation where, let's say, for example, you're exercising and you're 3 miles into a 5-mile run. Oh, your muscle cells are fermenting to keep you going. A number of your muscle cells are starving for oxygen. How do you know you're starving for oxygen? You can tell by your beating heart. You're sitting here right now, your heart rate is probably 60, 70, maybe 80. If you start running, it's going to shoot up because you're trying to supply oxygen to your working muscles to prevent fermentation. But it's inevitable. You will ferment. The longer you exercise, eventually you'll ferment. Unless you're an athlete and you exercise every day, you're conditioning yourself to ferment later. You see? You become an efficient machine. But for me, I'm fermenting standing in front of you right now. Just kidding. That was a joke, right? Bad one, maybe. All right. So, you have a situation where you don't have oxygen. The idea is the cell needs to survive. For me to continue to survive, I have to keep making these 2 ATPs. That means I have to keep glycolysis moving. Listen to it. It's critical that you understand that. I have to keep this one moving. And I learned in chapter 8, I think it was chapter 8, yeah, that if you build up the product can feedback inhibit. Remember that? If I don't get rid of pyruvate, it can go back and shut everything off. So even these 2 ATPs, I don't get anymore. And how will you know when that happens? When you drop dead. So at times when I don't have oxygen, at least I have these 2 ATPs to survive on. And then the cell is waiting, come on, oxygen, come on, come on. Oh, when the oxygen comes back, feels happy, it will do the other one. But at least temporarily, it's still alive. using the ATP that it produced during glycolysis, and it will keep making 2 ATPs, keep making 2 ATPs, because it's pushing pyruvate out of the way. It's getting rid of pyruvate. So this glucose that just came in will break down the 2 pyruvates. The pyruvates get fermented to lactic acid in us, giving room for another glucose to come in and do the same thing. So I'll keep making 2 ATPs, 2 ATPs, 2 ATPs, so I can stay alive until the oxygen comes back, hopefully within a minute and a half or so, because I don't survive longer than that. So if pyruvate builds up, hopefully now you realize the whole thing shuts down. Then even these guys I don't have. So the cell has a problem, has actually 2 problems. Problem number 1, I have to get rid of the pyruvate. Do you see that? Otherwise, it'll feedback inhibit. Problem number 2, I have to keep a supply of the empty trucks. If you want to move on Sunday, Sunday is your move out day. You call all the U-Haul truck companies, they all tell you, we don't have trucks. Then you're not moving on Sunday. Do you see that? If you don't have these, then the whole thing shuts down. Then you don't get these. So look at that. You have 2 problems. The cell decides, I'm going to solve it with 1 solution. Oh, wow, what an amazing thing. 2 problems. Problem number 1, get rid of the pyruvate. Problem number 2, make sure you have enough loaded, empty trucks, NAD+. Solution, combine both problems in one with 1 solution. Here it is, watch this. As I break down the pyruvate, I take these and use them and empty them. Do you see that? To break down the pyruvate, you're consuming the loaded trucks, producing unloaded trucks. So that when the next glucose comes in, these guys will go back here to produce these guys again. And it goes back and forth. I mean, again, if that doesn't fascinate you, you are hard to please. It's amazing. 2 problems, 1 solution. You're going to get rid of the pyruvate like you need to. And always, you will, because you link these guys to that, you will always have the empty trucks available all the time. So you get to survive. But again, don't push your luck, because for us, we need a lot of energy. So hopefully, oxygen comes on the scene real soon. But at least temporarily, this is what happens. So now, I'm going to keep the NADHs here, because now we're going to go into the situation, the scenario where oxygen is present. So the red lines will be describing to you the events when oxygen is present. I did not write on the wall, thank God. I need this board. So oxygen is present. So it's this one. What's going to happen? This is going to happen. Pyruvate oxidation. When oxygen is present, the pyruvate is going to oxidize. That means break down. Break down into what? Break down into acetyl-CoA. As it's entering the mitochondria. So I have to draw or show you a representation of the mitochondria. Got the outer membrane. The inner membrane is a little difficult to put on the board, but we'll deal with it. Let's see. Hopefully I have more success this time. Do it one step at a time. There we go. Good. All right. So follow the red line here. Good. All right. So the pyruvates, in the presence of oxygen, will enter the mitochondria. Obviously, we're talking about a eukaryotic cell, because prokaryotes do not have a mitochondria. Where does all of this happen in a bacteria? You'll deal with that in another class. We don't talk about it here. You'll deal with it in, let's say, microbiology or something like that. But we're going to restrict our discussion to eukaryotes. So the pyruvates will enter the mitochondria, and that's why I have two membranes. Remember, the mitochondria has two membranes. It has an outer membrane and an inner membrane. So that's the outer membrane. This is the inner membrane. It's folded, and I told you it needs to be folded, because these folds are actually very critical, because they create a space that's called the intermembrane space. That space is critical. So what happens is the pyruvate will come in and it will become acetyl-CoA. If you notice, it is now 2 carbons. Pyruvate is 3 carbons. It's now 2 carbons, acetyl-CoA. So what happened was, watch, the 3-carbon pyruvate became a 2-carbon acetyl-CoA. The 3-carbon became 2 carbon. What happened to this carbon? You're breathing it out right now. It's carbon dioxide. So, you're releasing CO2. But whatever happens to one pyruvate happens to the other one. So you actually make 2 CO2s. See, one pyruvate becomes acetyl-CoA, but you have 2 pyruvates. So you actually have 2 acetyl-CoAs. You actually have 2 of these. But we'll get back to that. But you have 2 CO2s. Each pyruvate will make a CO2. And then there are electrons coming off. There are electrons coming off. So, yeah, you need the empty truck. And you'll produce a loaded truck. For each pyruvate, you'll make one of these. That means you need another one for the second pyruvate. There it is. So if we tally things so far, without oxygen, we're going back to without oxygen. Without oxygen, we only had 2 ATPs. Even the NADHs we don't have. We use them, remember? Without oxygen, we only have the 2 ATPs. With oxygen, the NADHs don't get used up here. They actually go to the mitochondria. These NADHs produced in glycolysis end up in the mitochondria. So if we tally things so far, from glycolysis, we have 2 ATPs. made by SLP. And then we have two NADHs from glycolysis. From pyruvate oxidation, we have two NADHs and two CO2s. Here they are. Everybody okay so far? All right. Now, we're now in the region of the mitochondria called the mitochondrial matrix. Mitochondrial matrix. That's the location in the middle of the mitochondria. That's the matrix. The acetyl-CoA is going to bind with oxaloacetate. Acetyl-CoA with oxaloacetate. And they're going to make citrate. They're going to make citrate. Where are your citrate? Here. The 2-carbon acetyl-CoA binds with the 4-carbon oxaloacetate, making the 6-carbon citrate. The citrate is going to become isocitrate. Isomerizes, isocitrate. All right, this isocitrate will now become alpha ketoglutarate. The alpha ketoglutarate will become, where are you? Here it is. Succinyl-CoA. Succinyl-CoA will become, it's a cycle, you guys. It'll go back to oxaloacetate. Where are you? Right here. Succinate. Succinate will become fumarate. Fumarate will become, I know, malate. Don't blame me, blame yourselves. And malate will become oxaloacetate. All of that is happening in the mitochondrial matrix, and that is called the citric acid cycle. It's a cycle. So whatever it started out with, oxaloacetate, it reproduces. Otherwise, it's not a cycle, and the whole thing shuts down. Now, one of the most important things for you to know is what is being produced by this. We're only looking at one acetyl-CoA. We're going to go through the cycle once. For each acetyl-CoA, you turn the cycle once. But how many acetyl-CoAs do we have from glucose again? 2. So I'm going to show you one turn of the cycle, but remember, we have to multiply everything by 2. From this to this, you're putting things together. So you're going from 2 carbons, 4 carbons, putting them together, you got a 6-carbon citrate. That's done. It isomerizes. Good, we didn't lose any carbon. We're fine. Now, from here to here, look at that, 6 carbon, 5 carbon, we lost carbon. In the form of what again? Every time we lose carbon, what is it? Carbon dioxide. Carbon dioxide. On the outside are the things being produced. On the inside, I'm going to put the things being consumed. So CO2 is produced, of course. You're going to get rid of it. You just breathe it out eventually. All right. NAD plus, that's an empty truck, produces a loaded truck here. We go from 6 carbon to 5 carbon, we lost a carbon. Okay, we go from 5 carbon to 4 carbon, so we lose another carbon. We use another empty truck, produce a loaded truck. And then with respect to NADH, we're going to go all the way to the step between malate and oxaloacetate. Here we make NADH, use NAD+, but there's no CO2 production. This is 4 carbon, that's 4 carbon, so there's no CO2 here. So how many NADHs are made here? 3. But remember, we turned the cycle on twice. So we're going to double those. But before we go there, between succinyl-CoA, we use ADP, make ATP by SLP. Between succinate and fumarate, here comes the other truck. We use FAD, make FADH2. That's the only place we use this guy. All the other trucks are NADH. Do you notice that? NADH in glycolysis, NADH in pyruvate oxidation, NADH in the citric acid cycle. This is the only place that you use FADH2 or produce it. That's an awesome exam question. These we call electron carriers. That's what they're doing. They're carrying electrons. There are 2 kinds, NADH and FADH2. NADH obviously is used more frequently. FADH2 is only in the citric acid cycle. So, like I promised you, remember, we have to double things because we don't have just one acetyl-CoA, we have 2. So let's see if I got all of them. Let's see. So remember, it's actually 2 acetyl-CoAs. I didn't want to do more of those because it gets to be way too busy. It's busy enough as it is. Here it is. We have to double up here. We have to double up here. Double up here. Double up. This one. Everything is doubled. I'm missing these. So now if we tally things up from 1 glucose with oxygen, what do we get? Well, from glycolysis, 2 ATPs, 2 NADH. From pyruvate oxidation, 2 NADHs, 2 CO2s. Remember, we get rid of those. That's waste. By doing what? Breathing it out. From the citric acid cycle, which is also, by the way, called the Krebs cycle, in case you see that, they're discussing the same thing. Let's tally things up. How many CO2s do we get? 4. How many total NADHs? How many total ATPs? By SLP, remember? How many of the other truck? 2 of these guys. That's your accounting. But all of this, I only showed you 4 ATPs. 4 ATPs out of the 38 that I promised you. Very disappointing, right? Where's the other 34? You lied. No, they're coming. So the 4 ATPs that are made so far are made by SLP. 2 from glycolysis, the net 2 from glycolysis, and the 2 from here. 2 turns of the cycle. Now I have to show you the fun part. The fun part is, where are the other 34? That's when the delivery trucks come in play. And if we look at the delivery trucks and we add all of them up together, how many total NADHs do you see? The total number of NADHs from one glucose. 10. Here they are. 2, 4, 10. You're right. 10 NADHs total. How many total FADH2s do you see? 2. Only these 2. You're right. Here they are. The reason I put them this way with their unloaded truck is because of what they're about to do. What do delivery trucks do again? They deliver. In this case, what are they delivering? Electrons. These guys are about to deliver their electrons. To who? Hang in there. But once they do, they become empty trucks. Do you agree? Once you unload your furniture from your truck, that truck leaving your driveway is empty, I hope. So know that as soon as they deliver the electrons, they will now become empty. So they're going to deliver electrons. Well, it's the same thing with FADH2. As soon as they deliver electrons, they will leave empty. The question is, who are they delivering electrons to? Obviously, whoever it is lives in the inner membrane of the mitochondria, because that's where I'm pointing towards. Yeah, there are complexes in the inner membrane of the mitochondria, and they're called complex 1, complex 2. Can you guess the next one? Yep, 3. And 4. And then there's another guy. This is your buddy. This guy is called ATP Synthase. That's the guy that's making your ATP. They're all in the inner membrane of the mitochondria, sitting there. Of course, the membrane goes all the way around, so there's multiple ones. I just show you one. The idea is this. The ATP synthase I told you is your buddy, right? Because that's the guy that's going to make ATP for you. But the problem is, he's sleeping on the job. And someone needs to wake him up. Wake him up to do what? To make ATP. ATP synthase will literally do this, take ADP, a phosphate, and make ATP out of them. ADP, a phosphate, and make ATP out of it. Knows how to do that very well. But someone needs to wake him up, or something. It's hard to wake up. So sleeping on the job. All right. So how do we wake them up? Here's what's going to happen. The electrons from NADH will go to complex 1. We will first follow the electrons. Then there's something else. There's something called ubiquinone. And cytochrome C. Ubiquinone is usually like somewhere here, almost very close to 2, like somewhere, it doesn't really matter exactly where, but after 1, near 2. And cytochrome C is between 3 and 4. The electrons from NADH will go to complex 1. Then it's like a relay system. I want to give the marker to the person all the way in the back, but I don't want to go walk back there. So I give the marker to you, you give to them, and then it goes all the way back. Oxidation reduction reactions. So the electrons received by complex 1 will now go to ubiquinone, also called coenzyme Q. You can find that on the shelves near your vitamins. And then you go from coenzyme Q to complex 3. Who's doing that? The electrons. And then from complex 3 to cytochrome C. See, I can't draw it in. They're actually moving in the membrane, by the way. Don't think that the electrons are jumping in that space. No, I just can't draw it that way. The electrons are actually moving within the membrane here from one complex to the other. And then from complex C to from cytochrome C to complex 4. Now, something has to get the electrons from complex 4, because for complex 4 to participate in the next event, it cannot be caught with electrons in its hand. Watch this. If I'm complex 4, I have electrons. I can only handle 2 electrons. What do you want me to do? You want me to take 2 more? Yeah, and I got to get rid of these first. So I have to get rid of these first for me to take 2 more. Then I have to get rid of these for me to take 2 more. What would happen if I can't get rid of them? If complex 4 cannot get rid of the electrons that it got from cytochrome C, then the whole thing shuts down. It backs up. And no ATP will be made. You're in trouble. So the question is, who takes the electrons from complex 4? You're breathing it right now. It's oxygen. Oxygen. Here it is. Oxygen was delivered to the cell by the red blood cell. It went into the cell. It is small and non-charged. So it entered the cell how? Simple diffusion. So some of it is in the cytosol or cytoplasm. But then some of it reaches the mitochondria. Oxygen takes the electrons from complex 4. We therefore call oxygen the final electron acceptor. That is a guaranteed question on all my examinations from this point on. So, this is what's going to happen right here. The electrons from complex 4. Let's see if I can do a better line here. The electrons from complex 4 are these electrons. The electrons from complex 4 will go to oxygen. Along with hydrogen, it'll become water. So the cool thing is, the oxygen that you're breathing right now will become water at the end of cellular respiration, because that's your final electron acceptor. If you're not breathing, none of this is possible. The only thing that's possible is the 2 ATPs. So really what happens is pyruvate is made at the end of glycolysis, and it checks to see, hey, do we have oxygen? Yes, let's go that way. We don't have oxygen? I'm going to ferment. I mean, there's no way pyruvate will do all of this, and then surprise, there's no oxygen. Oh boy, you're in trouble. You're in trouble. So the decision-making process of whether to ferment or not is at the pyruvate level. Do I have oxygen? Yeah, I'm going to the mitochondria. Do I have oxygen? No, I'm going to do what? Ferment. Into what? Well, it depends on who you are. Animal cells, lactic acid. So, of course, the scenario we're talking about here is in the presence of oxygen. So you're breathing oxygen. Oxygen is the final electron acceptor. It gets the electrons and now becomes water. Good. Now, did we make ATP? We did not make ATP, right? We're only dealing with the electrons. But we have to deal with the electrons from FADH2. We dealt with the electrons from NADH, but we also have to deal with NADH2. Now watch this. NADH2 gives the electrons to complex 2. I think I harassed you about these, right? On the line application, I think it was your class, right? I harassed all my sections, by the way, right? So I asked you guys, who does NADH give the electrons to? Eventually someone said complex 1. You're right. Who does FADH2 give the electrons to? It's complex 2. But from that point on, the same events occur. Same thing occurs. So look at this now. Watch this. NADH uses complex 1, 3, and 4. NADH uses complex 1, 3, and 4. FADH2 uses complexes what? 2, 3, and 4. But watch this. Number 2 does not do something that the other ones do. Watch this. These guys, when they get electrons, they pump hydrogen. So as soon as they get an electrons, look what they do. They pump hydrogen into this space. That's what this space is for. Complex 1 pumps hydrogen. That's why I wrote it up here. I'm only writing the complexes that actually pump hydrogen. 1, 3, and 4. When NADH is involved, it's 1, 3, and 4 that are pumping hydrogen. 1, number 3, and number 4. So 1, 3, and 4 pump hydrogen. Number 2 never pumps hydrogen. Number 2 takes the electrons from FADH2, but it does not affect it at all when it comes to hydrogen. So only 3 and 4 are pumping when number 2 is involved. So NADH causes hydrogen ions to pump from complexes 1, 3, and 4. It doesn't deal with 2 anyways. FADH2, yes, it deals with 2, complex 2, but complex 2 doesn't pump hydrogen. So only 3 and 4 pumps hydrogen. So there's a lot of hydrogen in the space, and there's a lot less inside. I told you I'm going to challenge you with this before, and here comes. What is that called? Say? A gradient. What kind of gradient? You're absolutely right, and I heard it from here too. That's exactly what it is. That's your electrochemical gradient. It is a gradient, a distribution of charge across the membrane. A distribution across the membrane, that's a gradient, but now that's something charged. It's an electrochemical gradient. That's your battery. Remember that? Now this battery, guess what it's designed to do? It's designed to wake this guy up. Remember that guy? You forgot about him, right? I told you we got to wake him up. The electrochemical gradient, the battery, is going to wake this guy up. And he's hard to wake up. You almost have to, not almost, you actually do. You have to force the hydrogen through it. This is actually, it's better if it's oriented this way, but it's okay. That's a minor detail. The hydrogen will be actually forced through it. you pump the hydrogen back through it, back into here. So the hydrogen will eventually be pumped back here. And at least some of this hydrogen is what's doing this. At least some of this hydrogen will be used for that. It's the pool of hydrogen. It doesn't matter what it's used for. But what matters more importantly is that it pumps through here, waking this guy up. Waking this guy to do what again? Grab an ADP, a phosphate, and make what out of it? ATP. Did I put that on the screen? Here it is, yeah. That's this guy. That's what this guy is doing. ATP synthase is going to grab an ADP, a phosphate, and make ATP. I think you're used to that by now. Everything has a name. That's called chemiosmosis. These guys, it's called the electron transport chain. That's these guys. Complex 1, 2, 3, and 4. Together, it's called oxidative phosphorylation. The electron transport chain plus chemiosmosis, which is this, is called oxidative phosphorylation. So the CO2 that you make, how does it leave the cell? It's small and non-charged. Simple diffusion. So all the CO2 that you make will eventually find itself near the membrane and go right through it. So CO2 is leaving. Oxygen is, of course, coming in by simple diffusion. Who's delivering the oxygen? The red blood cells. Who's picking up the CO2? The red blood cells. It's amazing. They deliver your oxygen and they pick up your garbage at the same time. And the garbage in this case is CO2. So now, who creates a stronger battery? NADH or FADH2? NADH. Why? Because there are 3 complexes involved in pumping hydrogen. FADH2 creates a battery, but it's a weaker battery because only 2 complexes are involved. It's all about the hydrogen. So as it turns out, 1 NADH will make 3 ATPs. 1 FADH2 will make 2 ATPs. Your book tells you otherwise. Your book says 2.5 and 1.5. And they justify the reason because, at least partly, it takes energy to move pyruvate into the mitochondria. The 2 NADHs made here, they have to come into the mitochondria. That's like living in Florida. We know, we're used to that. You use the highway, you better have money, right? That's toll. That's your toll charge. It costs a little bit of energy to bring in the pyruvate, to bring in an NADH. Sometimes the NADH wants to come in as NADH, but then it turns into FADH2. Don't worry about the detail here. What all you need to worry about is it takes a little bit of energy to bring these things from the cytoplasm into the mitochondria. That's what I call your bank fees. So you might get paid $100 on Friday. But if your bank charges you $2 to keep the money in there, you'd be foolish to walk around saying, I have $100. You actually have 98. Those are your fees. But now it all depends on which cell and all of the. So listen to me. I teach my class based on this. 38 ATPs maximum. That's what you're going to get. The actual is variable. You're done. Well, I'm not going to teach you all the different possibilities here. I show you that the maximum yield is 38 ATPs. Know that, yeah, that's it. But the actual is variable. It depends on the bank fees. So I bank at Wells Fargo. You bank at whatever. What else? What's another name for a bank there? Chase. Yeah. My bank fees is $2. Yours is $4. My $100 become 98. Yours becomes 96. Do you see? So what I'm going to do? Teach you all the bank fees? No, no. I tell you, you get paid $100 on Friday. That's it. What happens to it is technically a little bit irrelevant at this point. So I'm teaching you based on maximum yield. It's 38. Your book factors in the bank fees. In case you go back to your book and say, wait a minute, he lied to us. I know, no. If you read the fine print, they'll tell you that. So, let's finalize it. We have 4 ATPs. 1, 2, 3, 4. Where are the other 34? Well, you had 10 NADHs. Each NADH makes 3 ATPs. So 10 of them makes how many? Yeah. You have 2 FADH2s. Each one of them makes 2. Now you have what? $30 plus 4 is 34. Plus 2, 36. Plus these 2. Plus 2 more is 38. So a good exam question would be, how many out of the 38 ATPs are made by substrate level phosphorylation? How many? How many? 4. Another question is, yeah, okay, 4 are made by SLP. The other 34 are made by what process? Oxidative phosphorylation. And when you say oxidative phosphorylation, you're saying both of these at the same time. What do you guys think? You got your work cut out, but you can do it.